Hello, everyone. Welcome back to Meds.Concepts. Today we will discuss an important topic of molecular biology and epigenetics, regulation of chromatin structure. Chromatin is not simply a packaging material for DNA. It is a highly dynamic regulatory platform that controls gene expression, genome stability and cellular identity. In eukaryotic cells, DNA is wrapped around histone proteins to form nucleosomes, but this chromatin organization is continuously modified according to cellular requirements. When chromatin is open and accessible, transcription factors and RNA polymerase can bind DNA and activate gene expression. In contrast, condensed chromatin restricts DNA accessibility and represses transcription. Therefore, chromatin regulation acts as a molecular switch controlling cellular functions. Chromatin remodeling complexes. One major mechanism of chromatin regulation involves ATP-dependent chromatin remodeling complexes. These are large protein complexes that utilize ATP hydrolysis to reposition, eject or exchange nucleosomes. The major remodeling families include SWI-SNF, ISWI, CHD and INO-AT complexes. SWI-SNF complexes are especially important because mutations in their subunits are associated with many cancers. Chromatin remodelers can slide nucleosomes along DNA, remove histones completely or replace histone variants. Through these mechanisms, remodelers regulate DNA accessibility for transcription, replication and DNA repair. Mechanism of nucleosome sliding. Now let us understand how nucleosome sliding occurs. First, ATP hydrolysis activates the remodeling complex. The remodeler then functions as a DNA translocase and moves DNA directionally around the nucleosome. This movement creates a small DNA loop on the histone surface. The loop gradually propagates around the nucleosome, causing repositioning of the nucleosome along DNA. As nucleosomes move, promoter and enhancer regions become exposed or hidden, thereby regulating transcription. Regulation by affecting nucleosome binding. Nucleosome positioning is strongly influenced by DNA sequence and DNA binding proteins. At-rich DNA sequences bend more easily around histones, while GC-rich regions are generally less flexible. Certain DNA sequence patterns therefore favor nucleosome formation. DNA binding proteins can also block nucleosome assembly nearby and create nucleosome-free regions. Such nucleosome-free regions are commonly found at promoters and enhancers where transcription factors need access to DNA. Thus, chromatin accessibility is tightly controlled by nucleosome positioning. The core histone octamer. At the heart of every nucleosome is the core octamer. As you can see in the diagram, it is composed of two copies each of H2A, H2B, H3, and H3. These proteins are the spools around which DNA wraps. The linker histone, H1. It plays a critical role in stabilizing the chromatin fiber and helping it fold into more compact structures. Histone variants and their functions. Beyond the standard histones, the cell utilizes histone variants to change the property of chromatin in specific areas. Let's look at the key ones mentioned here. H3.3. This variant is primarily associated with transcriptional activation. It replaces the standard H3 in regions where genes are being actively read. CENPA. This is a specialized H3 variant found only at the centromere. It is essential for assembling the kinetochore and ensuring chromosomes separate correctly during division. H2A.X. This is a crucial variant for DNA repair. When DNA breaks occur, H2A.X is phosphorylated to signal the cell's repair machinery to the site of damage. H2A.Z. This variant is versatile. It is involved in both transcription regulation and gene silencing, depending on where it is placed in the genome. Histone modifications. Histone tails undergo multiple post-translational modifications known as PTMs. These include acetylation, methylation, phosphorylation, and ubiquitination. Together, these modifications create the histone code, which determines chromatin structure and transcriptional activity. Acetylation generally activates transcription by loosening chromatin structure. Methylation may either activate or repress genes depending on the modified residue. Phosphorylation is involved in mitosis and DNA damage signaling, whereas ubiquitination participates in DNA repair and transcription regulation. Thus, combinations of histone modifications determine cellular outcomes. Histone reader domains. Histone modifications exert their effects through proteins containing specialized reader domains. Bromodomains recognize acetylated lysine residues. Chromodomains bind methylated lysines. Tudor domains recognize methylated lysine or arginine residues. PhD finger domains often recognize H3K4 methylation, which is associated with active chromatin. Some domains interact with unmodified histone tails. These reader proteins decode histone marks and recruit regulatory complexes to chromatin. Histone chaperonase and nucleosome assembly. Nucleosome assembly is not spontaneous and requires histone chaperonase. Histone chaperonase prevent inappropriate histone aggregation and guide histone deposition onto DNA. Important histone chaperonases include CAF1, ASF1, FACT, and NAP-AIC. CAF1 deposits H3H4 histones during DNA replication and interacts with the replication factor pCNA. ASF-AIC transfers H3H4 dimers, while FACT facilitates transcription through chromatin by temporarily destabilizing nucleosomes. Thus, histone chaperonases ensure accurate nucleosome assembly and chromatin maintenance. X chromosome inactivation. In female mammals, dosage compensation is achieved through X chromosome inactivation. This process is initiated by a long non-coding RNA called XIST-RNA. XIST coats one X chromosome and recruits chromatin-modifying proteins. The inactive X chromosome accumulates repressive marks such as H3K27 methylation and DNA methylation. Histone decetylation also contributes to chromatin condensation. Eventually, the chromosome forms highly condensed facultative heterochromatin called the Bar body. Thus, X chromosome inactivation is an important epigenetic phenomenon. Histone methylation and phosphorylation. Histone methylation occurs mainly on lysine and arginine residues. Some methylation marks are associated with active chromatin, such as H3K4 trimethylation and H3K36 trimethylation. Other marks like H3K9 trimethylation and H3K27 trimethylation are linked with transcriptional repression and heterochromatin formation. Histone phosphorylation occurs on serine, threonine and tyrosine residues. Phosphorylation of H3S10 is strongly associated with mitotic chromosome condensation. Additionally, phosphorylation participates in DNA damage response signaling. Chromatin and DNA repair. Chromatin structure changes rapidly in response to DNA damage. DNA double-strand breaks trigger chromatin relaxation around the damaged region. One of the earliest events is phosphorylation of H2AX, producing H2AX. This modification recruits DNA repair proteins such as BRCA1, RAD51 and repair signaling complexes. ATP-dependent chromatin remodeling complexes also facilitate access of repair machinery to damaged DNA. Therefore, chromatin modifications are essential for efficient DNA repair. Epigenetic inheritance. Epigenetic information can be inherited during DNA replication. During replication, parental H3H4 tetramers are randomly distributed to daughter DNA strands. New histones are then incorporated alongside parental histones. Importantly, parental histones carry pre-existing histone modifications, which help restore chromatin states after replication. This mechanism maintains epigenetic memory across cell divisions. Therefore, cells preserve stable gene expression patterns even after DNA replication. Experimental techniques to study chromatin. First is ChIP-seq. This is the gold standard for mapping specific protein-DNA interactions. By using antibodies to pull down specific proteins, like transcription factors or modified histones, we can sequence the attached DNA to see exactly where these proteins are binding across the entire genome. Next, we have ATAC-seq. This technique uses hyperactive enzyme called TN5 transposase to tag and sequence regions of open or accessible chromatin. It tells us which parts of the genome are active and available for regulatory proteins to bind. To understand the physical spacing of nucleosomes, we use MNS-seq. This method uses an enzyme that digests the linker DNA between nucleosomes, leaving only the DNA wrapped around the histone core. This allows us to map the precise positions and density of nucleosomes. Next, to see the big picture, we use Hi-C. While other methods look at linear sequences, Hi-C captures the 3D architecture of the genome. It reveals how DNA loops over long distances, bringing distant enhancers into contact with gene promoters through topologically associating domains, or TADs. Moving to the right side of the slide, we have imaging and microscopy techniques like STORM and 3D FISH. While sequencing gives us data, these tools allow us to actually see the chromatin. They provide high-resolution images of how chromosomes are spatially organized inside a single cell and help us measure the physical distances between different genomic regions. Finally, we have nanopore sequencing. This is a revolutionary tool that sequences long strands of DNA in real time by passing them through tiny pore. The unique advantage here is that it can directly detect DNA methylation and other modifications without the need for complex chemical treatments, giving us a clearer picture of the epigenetic landscape as it exists naturally. Chromatin dysregulation in diseases. Defects in chromatin regulation contribute to many diseases, especially cancer. Mutations in chromatin remodeling complexes such as SWI/SNF are common in several tumors. Rett syndrome is caused by mutations in MECP2, methyl-CpG binding protein. Coffin-Siris syndrome is associated with defects in chromatin remodeling components. Epigenetic therapies are now used clinically. DAC inhibitors and DNMT inhibitors are examples of drugs targeting chromatin regulators. Thus, understanding chromatin biology has major medical importance. Finally, let us revise some important concepts. Nucleosome remodeling is an active, ATP-dependent process. The histone code relies on reader domains to translate modifications into biological actions. Key markers to remember: H3K4 trimethylation signals active promoters, while H3K27 trimethylation indicates repression. Similarly, DNA methylation at CpG islands ensures stable gene silencing. For regulation, polycomb maintains repression while trithorax maintains activation. During replication, CAF1 deposits histones via pCNA-dependent path, and XIST-RNA drives X inactivation. Finally, H2AX marks DNA breaks, and epigenetic inheritance ensures these states persist through cell division. Thank you. If you found this video helpful, please like and subscribe for more.